So that leads us here into the replication of DNA. DNA replication is the process. Let me get my little coloring pen here. Is the process of copying a DNA molecule. Okay. Copying a DNA molecule. It is what we refer to as semi-conservative. Okay. So if this is the DNA molecule unwound, okay, when it gets replicated, I'm going to replicate this and that. Okay. So there's my parental one. And then I'm going to do another color here. Let's do green. Then there's a copy. And there's a copy. And I still have my double-stranded helix structure because it'll wind back up. Okay. But it is semi-conservative. I still have half of the original. Okay. Each strand of the original serves as a template. And we get a new daughter strand with it. And the daughter strand would be the green one here. Okay. It would be the green one. Green. Okay. Here's a picture that maybe shows it off a little bit better. There you can see it right there. Okay. It unwinds. Doesn't unzip from stem to stern, but it just unwinds a little bit in a spot. And we copy them. Okay. And it's semi-conservative, one original and one new. And then that'll be the new one. And then we'll have one original and then one new. And then this will be one original and one new with it. So it's semi-conservative. Okay. This unwinding of this DNA strand is due to an enzyme. And it's underlined. And should maybe remember that. Because we can copy this. We can make DNA a helicase. Okay. The DNA helicase is an enzyme that I can make, and through magic, I can do this in a lab with any DNA that I want to copy, that I want to replicate. All right. And I can do that with this thing called DNA helicase enzyme. Then I'm going to bind the complementary base pairs, and that's what single-stranded binding proteins, that's what it's talking about, okay? Binding, ING, binding complementary base pairs. Okay. Again, we have DNA helicase, which opens up the DNA helix. I have DNA primase that allows me to attach these complementary base pairs in there. And ultimately, it'll get sealed back up, and I have replicated my DNA.
DNA replication will begin at a specific area of the molecule called the origin of replication. The origin of replication denotes the area of active replication called the replication fork. In order to understand how complex eukaryotic organisms replicate DNA, scientists first studied replication in prokaryotic models like E. coli. A number of enzymes are needed for replication to proceed once the replication fork is established. Helicase separates the strands of the double helix, and single-stranded binding proteins stabilize the newly single-stranded regions. DNA gyrase is used to make sure the double-stranded areas outside of the replication fork do not supercoil.
So you can see there's a lot of stuff going on here. And I'm trying to make it as simple as possible. And I know you're probably sitting there going, you're a liar. And I'm not a liar. I'm just trying to keep it simple. Okay. Because if I don't have some of those extra enzymes that I didn't talk about, this thing will just uncoil, or it'll coil too much because it's trying to retain its structure. So it's going to just reflexively try to coil back down on itself to stop itself from unwinding. And so these enzymes help to just keep this replication fork, these kind of replication bubbles, if you will, from keep it stable and prevent it from, from the structure of it from just falling apart. When we look at a eukaryotic organism, which we'll talk about mostly here, it'll have these little replication bubbles in multiple different spots. How does it figure that out? We'll get to it, but spoiler alert, it's this genetic code. It's these sequences of nucleotides that actually say, hey, start here, okay? Because that's what I code for. It's start. In a prokaryotic organism, it just has a circular loop of DNA. So it just starts on one end, boom, goes all the way around. And as you can see, it doesn't take too hell of a long time, okay? To replicate their DNA takes, maybe 40 minutes. They divide every 20. So you can see by the time I get a second generation, it may not even be done replicating its DNA yet. The significance of that is, remember the nucleoid, the DNA material is in a nucleoid region here. So remember, it's just kind of this kind of cloud-like material of DNA all congealed in the center aspect of it. To get it to precipitate down and form these circular rings, these circular shaped chromosomes that they have, it's much more simpler and complex than it is when it comes to the eukaryotic organism. And we saw some of that when we looked at mitosis, binary fission, right? Where we looked at how DNA condenses down and replicates. In a prokaryotic organism, super simple, super fast, okay? Some of their generation times are 15 minutes. So we can get a fast amount of DNA being replicated in a number of various different bacteria. And some bacteria may not have a full complement of genetic material, but they replicate so damn fast, it doesn't really matter. So we're going to look at eukaryotic DNA replication here, okay? This is where it begins at numerous points because DNA is much more complex, much more longer than a prokaryotic organism. If we started on one end and had to go all the way down the SOB, that would take forever. So we can unwind and unzip this DNA at multiple different locations and copy, replicate the DNA. Again, it requires an enzyme for that, and that's that DNA polymerase. DNA polymerase is going to allow us to the DNA helicase is going to open it up. DNA polymerase is going to allow me to copy this thing fairly quickly. Okay. This is prokaryotic, and this is eukaryotic organism right here. So here it just kind of starts and goes around, and that's what you're seeing right here. Here on a eukaryotic organism, you can see you can have a multitude of various different spots where it starts, all right? And this replication bubble just extends down in both directions, and that's what they're saying right here. The replication bubble spreads bidirectionally, okay? And again, it's semi-conservative. We have telomeres at the end to keep everything stabilized. We talked about that in chapter 9. So this can happen at a multitude of various different locations, not necessarily just two, but a number of them, okay? And there's my DNA helicase unwinding. There's my DNA polymerase going down and copying them all, okay? Remember, one goes 5' to 3' really easily. The other one, remember, it's the opposite. So it has to go backwards, and that's what they're trying to represent down there. And I am not in no way in hell going to go into Okazaki fragments and how it's the lagging strand and how it goes from 5' to 3', but 5' to 3', it reverses itself. So, la, 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 la. DNA gets copied fairly easily from the 5' to 3' direction. Boom. And then don't even look at that last little strand right there. Okay. The great thing about this DNA polymerase and copying, and it's got to ensure that it gets all these complementary base pairs together there. It does a really fantastic job of ensuring that it copies it correctly. I mean, you can see it's not foolproof, okay? But it does have the capability of knowing that it done messed up, and it can go in there and actually cut out the misread nucleotide and correct itself. So that is the most fabulous thing about our DNA, right? We talked about these checkpoints in the cell cycle where it checks the DNA, and here's an homage to that in chapter 9, okay? So this just further proves that even though it does screw up from time to time, okay, it can go in there and cut it out and fix it and make it right.